that was an adventure. I'm glad we're all here. I apologize for the delay. Thanks everybody for being here, and thanks Chris and Debbie for the very kind introduction, which we all heard, I guess, a good while ago now. And I'm going to be talking today about putting single cell statistics into biological context and how we make sense of some pretty big data. Uh-oh, that should have made my slides advance. Okay, there we go. Okay, so all of the cells in your body share the same DNA, but they have wildly different functions despite this shared DNA. And what we're trying to understand when we do RNA sequencing or when we do the kind of biology that I do is how those functions change in different contexts, in different diseases, et cetera, and then use those changes to predict treatments or help people recover from diseases and that sort of thing. So when we talk about cells having different functions and just being different, what we mean is that they might have a different appearance. So historically, when we talked about cell biology, we were talking about what cells looked like under a microscope. And then we also think about their surface markers or what proteins are on the outside of the cell. And those surface markers, I call them markers, but really they're like receptors for the most part. So they're listening for chemical signals outside of the cell. And which signals cells are responding to tells us something about what those cells are doing. But the cell is also all of the different proteins that are inside of it, and all of those are playing their own roles. So when we think about what makes a particular cell different from another cell, we're thinking about what are the different proteins and the different components of that cell that make it different. So in an ideal world, what we would like to be able to do is measure the cell's protein content. So be able to like take an individual cell, blow up like, okay, this had this many of this protein, this many of this other protein. It's listening along these axes and like sending signals along these other ones. But unfortunately, protein on its own is really hard to measure, particularly for individual cells. So to get around that, we take advantage of what's called the central dogma of biology. And this is the idea that within your DNA, it encodes all of the proteins that are part of the cell. And to get from the DNA to the protein, you make an RNA, which is just a piece of the DNA encoding one particular piece of protein. And we know what the code is, so we're able to read those RNAs out, and it tells us what proteins the cell is currently making. So even though knowing what proteins are in a cell might be difficult, we can instead read out the unique set of RNA for every cell and use that to define what each cell is, or define what we call a cell state. So experimentally, it's actually possible to get out single cell RNA sequences for thousands or even now tens of thousands or more cells. And we do this using microfluidics. So we flow in, oh wait, we did have a laser pointer. Hey, okay. So within a microfluidic chip, we can bring in single cells along one entrance and tag beads. So these are just like tiny, tiny beads that are labeled with unique sequences of RNA. And then we make an emulsion similar to what you get in a salad dressing that contains just one cell and one bead. And once you do a bunch of chemistry to like pull the little pieces, the unique pieces off of the bead, you can collect the RNA from each cell tagged with a unique label. We would pool a bunch of these together and run sequencing. And then once we read out what those sequences are, we can map them onto what we know about the human proteome and be able to say what the cells are doing in our system. Okay, so that was like a big biological advance. And what it gets us is we can now go from a biological system to a big matrix, basically, of a set of cells, and within those cells, what their genes that are currently being expressed are. So this is an example of a duct from a human breast. And this is going to be, I guess, I'll talk about breast cancer later in the talk. So this is kind of the biological framing we're using. So in this particular case, we have all of these cells that we've read out. And then what we want to do is map those cells onto, or map those sequences onto individual cells. So then we try to label what the cell types are. And then from that, we then say what these different cell types are actively doing. So what RNA they're expressing tells us something about what their functional roles are. And we map those onto known cell types. So these might be immune cells, they could be epithelial cells, they're just different parts of the tissue. And then in the case of cancer or some other disease, we have different cell types and different cell states. So in this case, I've done in green are cancer cells. And it's immediately obvious that there's different numbers of some of these cell types. So there's all of these cancer cells here that weren't present in the non-cancer state. But there's more immune cells present that might be reacting to the tumor. And then the other question is whether the cells that are of a matched cell type are having a different functional role that they're playing. So are they in a different cell state with its own set of RNA? And is the RNA that's different something that matters for the system? So even though like the tumor is present, these immune cells or the other cells around it might also be doing something different, and we want to understand all of that. So can someone admit people on the Zoom room? Thank you. Okay. Yeah, so what we're trying to ask is what distinguishes different tissue states, and we ask that using the RNA content from single cells. And what I'm trying to do mathematically for this first part of the talk is think about how we quantify distinguishability, and within cells of a given matched cell type, can we say something about whether or not the cells are different between someone who does and doesn't have breast cancer, for example. So yeah, for the first part of the talk, I'll talk about distinguishability, and then I'll move from there into once we do have a defined set of distinguishable cells, what can we say about how cells of different types that are different between different systems are working together to get stuff done. Sorry, I'm going to take a second to admit people because I think it's not showing up. Or can you? Okay, thank you. I'm still seeing Etkos. That's my aunt's try. There we go. Okay, great. Thank you. Okay, great. So this first part of the talk on distinguishability is work that I did jointly with Stefan Peidley, who is a fantastic graduate student, or I guess now he's a postdoc, but he was a graduate student when we did this work. So we were advised, of course, by Chris and Debbie, but also by Stefan's advisor, Niels, and Augustin, who was formerly working with Chris. So that was the team on this project. Okay, so I've just told you, we get all of this beautiful single cell information, and then we can say what all of our cells are doing and what's all present, but it's a lot harder than I'm making it sound, which is, I guess, why they let me do an entire PhD in how to analyze the data. One of the reasons for that is that it's got a lot more dimensions than we have similar cells for the most part. So your genome has 20,000 or more genes in it. So we get out matrices for each cell that are or we get a vector for each cell that's 20,000 genes long. And it's also mostly zeros. Most cells are not expressing most genes, and the measurement method that we use is really noisy. So we miss a lot of what's actually present in each cell to begin with. So we've got what's called a sparse high-dimensional data matrix to begin with. And then on top of that, we have over-dispersed or not data. And what that means is that it's not Gaussian necessarily. So some statistical methods that we would use on typical data just aren't going to apply in the system. So we have to be careful with what we do in terms of our mathematical analysis. But at the same time, we still want to know how states distinguishable and how different are different groups of cells. And that's what I'm trying to ask here. So to think about how we answer this, I want you all to just take a minute to picture an n-dimensional sphere, which I know that's a big ask. So we'll start with a one-dimensional sphere, which I know is a lot easier. So that's just a line. And what we're thinking about here is how far apart are all of the points within this line. So for a single line segment that we're uniformly distributed along, they're uniformly far apart. So the portion of the line that's at the top and the bottom is just a pretty small amount of the overall line. But then if we start thinking about spheres in 2 dimensions, so that's just a circle, the fraction of your circle that's on the surface is larger than the fraction of your line that's at the top points. And if we think about a 3-dimensional sphere, the fraction of it that's on the outside of the sphere is again larger than the fraction of the area of a circle that's on the top there. So mathematically speaking, this is 2 over r is our surface area to volume ratio. This is 3 over r. I would love it if when we just kept adding dimensions, it's 4 over r, 5 over r. It's a little more complicated than that. But you'll have to trust me, this is a monotonically increasing function, meaning that as we increase our dimension, more and more of the volume of our space is really far away from the center. And what this means is that distance measures in high dimensional space don't make intuitive sense. What did I do? Okay. And just to really drive that home, this is a simulation that Stefan did, where we took spheres of increasing dimensions or uniform distributions on like a flat segment. And then we look at how far apart the points are within that. And in our one-dimensional system, it's very flat. And then as we increase our dimensions, we end up with a peaked distance. So as our dimension gets really high, all of the points in our data set are basically as close to each other as any other point. So just calculating a Euclidean distance in 500 dimensions, like, just doesn't make sense. So to get around this, the convention that we do is to reduce our dimensionality using PCA, but there's other ways you can do this. I'm just going to talk about PCA today. So PCA stands for Principal Components Analysis. And this is a method for reducing the dimensionality of our data by picking just directions that have the most variation. So this is just a two-dimensional case. And the first thing we have to do is center and scale our data. So different data sets might have points that are more spread out or less spread out or really big or really small. So we set them so that the average data point is at 0 and nothing is too terribly far apart from each other. And then we try to find the line in space that maximizes the variance of our data if we project it onto that line. So that means for each of these points, we draw a perpendicular onto whatever line that we've drawn, and then we calculate the variance of the points along that line using our standard variance equation here. Our x bar is going to be 0 because we've mean centered already. So that means that this is equivalent for those of you who get excited about linear algebra, which might just be me, but I get really excited about it. So this is equivalent to optimizing the transpose of x multiplied by x, where we've projected it onto some vector u, where u has the dimensions of our data set, and u is unitary. So it's if we multiply it by itself, it's equal to 1. So what that means is that the size of our data doesn't change when we do the projection, not the dimension, but like how big it is, I guess. And this thing here is equivalent to the variance. So I've made you look at that because we're going to come back to it a lot later in the talk. But the big thing to remember is that PCA is giving us smaller dimensions where the dimensions contain a lot of information. So that's what we try to do here. And when we've projected, we get a new data matrix, which is cells by principal components, by multiplying our PCA weight matrix, where this is our first U, and you just keep repeating this to get more U's out. Okay, so I've just told you a lot of math. Now we're going to go from that and try to use data that's been reduced in dimensionality to try to tell us how far apart our cell states are. And we're asking the question, how distinguishable are these? So this is just a sample where we have 2 different cell types or 2 different sets of data. And we're asking how distinguishable are these 2 clouds? And one natural way to think about this would be to look at how far apart the means are. So this is, we would take the average distance or the average location within each of our 2 clouds and then take the distance between them. But the problem with that is that there's a lot of distributions that have the same distance between the mean that I personally would consider much more distinguishable. So if we look at these 2 distributions, this looks by eye even much more distinguishable than this one. But if we just use the distance between the means, that's totally gone. Here's another few examples where these have the same distance between the means, but like this looks more distinguishable than these do. And we would like a measure which incorporates that information. So Stefan and I brought to bear on this problem something called the energy distance, which has been used a little bit for single cell, but we did it with, I guess, more depth, and I'll talk a little about that later. So energy distance is a measure of the ratio between how far apart on average points are in the 2 separate clouds relative to how far apart they are with between the 2 clouds. Oh no, please don't try to update my software right now. Oh gosh. Okay. We're back. Okay. Yeah, so delta here is the point-wise average distance between the 2 clouds, and then sigmas are the point-wise average spreads of the 2 clouds separately. And the difference between those is what we define as the energy distance. And And let's try calculating it just to make sure we're all on board. Here's our two sample point clouds. We have our red cloud, we've got our blue cloud. Let's go. Okay, so first let's look at our sigmas. So this is the average length of these red lines between all of the points in red would be our sigma x. Average length between all of the points in blue are our sigma y, and we calculate that using this equation. And one thing I want to note here is that the energy distance as was defined by Seckley and has been used historically is this here, which has 1 over n squared. And we introduced a bias correction term as part of our work. And this is because in the low cell regime or in the low sample regime, this inflates the distance artificially. So as you can see here, the value starts rising if we use this bias uncorrected point. And the reason for that is that if we look at each of the little clouds, if we don't use 1 over n minus 1, then we're adding in the fact that from each of these circles, there's a line like to that circle that has distance 0 always. So to remove that, we added this n over n minus 1. And this means that technically speaking, this is an unbiased estimator for the energy distance of two sets of clouds. So it's not actually a distance anymore, but it gets better performance on real data and on our simulated data too. Okay, so that was our sigmas. To calculate our deltas, we take basically the same thing, but now we're doing the distances between the two clouds. And that's our energy distance. So we take our two deltas minus our two sigmas, and that gives us a value. So let's think about which of these is going to have a higher energy distance. These two examples have the same delta, but our sigma for one of our clouds is smaller on this guy than it is on this guy. So a smaller sigma means a larger energy distance. So this is going to have a larger energy distance. Similarly here, our deltas are the same. And here our sigma is larger because we've got this vertical spreading here. And that means that this one is going to have a larger energy distance. So that's the general idea here. Okay, so once we have our measure of how distinguishable something is, we can use that to create a statistical test and have a number that comes out of how certain we are that two things are different from each other, not just how far apart they are. So for that, we use what's called a Monte Carlo permutation test. So we permute the labels of our data. So we've randomly assigned which one counts as red and which one counts as blue. And then we're going to go ahead and recalculate our energy distance. So we calculate delta again. And as we can see, more of our delta lines are shorter now. So our average delta is a little smaller. And we calculate our sigma again. And our sigmas now are much larger because we have these lines running across between the two clouds. And as a result of that, our energy distance is going to be smaller for our permuted data than it is for our real data. We repeat this well, probably less than 1,000 for this many points, but we repeat it a lot of times. And then we calculate the distance for all of our simulated data to create a null distribution of what the e-distance would be if we didn't have labels on our data, so if our data was random. And then we compare that to the value that we got using the actual data labels. And in this case, that's higher than our simulated values. So we would deem this to be statistically significant. So we do this by calculating a p-value, which is the number of e-distances on the right side of the red line divided by the total number that you calculated. So how many permutations we did. So when I talk about an e-test result, this is what we're doing. Okay, so we applied these to some real data sets. So just to build a bit of intuition here, this is a data set where people used CRISPR-A to perturb various genes. So that means the genes that are listed here are artificially activated. And then they went and sequenced cells and look at how activating these genes impacts the overall transcriptome. And what we've quantified with the e-distance is how much the cell state changed as a result of these individual perturbations. And as we can see in our low e-distance regime, cells don't move very much. So this is just a 2D projection of our data. And these just look like a big mush because there's not a lot of change. And we can compare that to permutations that had a much larger e-distance, where visually there's a lot of separability here. And the other thing we might notice is that some of these are looking close to each other, even though they're both far from the perturbed state. And we can actually use e-distance to quantify that too. So this is a different data set where the genes were interfered with rather than activated. And what we've done here is we've calculated pairwise e-distances between all of the transcriptional states when each of these genes is perturbed. And then we've clustered them hierarchically. So this group here is all different genes that were perturbed that had a similar cell state produced because their distances are close to each other. And these genes are all part of the JAK-STAT pathway, which is illustrated here. So this is a pathway where IFNG comes in, it triggers this cascade of signals, which then activate this protein, IRF1, which triggers a bigger transcriptional change in the cells. And if you knock out any component of this pathway, it creates a similar cell state because this IRF1 is interfered with in the same way. And with the e-distance, we can really easily show that visually. Okay, so I've just told you this is so great. We can draw all these nice pictures and it's very conceptually elegant. But to really say that something works scientifically, we have to do more than just argue that it makes sense. We need to really test it in a lot of different contexts. So to do that, Stefan and I, along with some Judy and some other people, we made a database of 44 single cell perturbation data sets of varying kinds of data and varying kinds of perturbations. And we annotation harmonized them so that they're you can computationally access all of them in the same way very easily. And then we use that to test our energy distance. And we've also made it publicly available as a database so people can use it to do their own experiments and their own modeling. And we've already had 4,000 data downloads from this. It's been up for about a year, or that was 4,000 a couple of weeks ago, so it's probably higher now. And it's pretty cool that people are actually out there using our little database that we spent quite a lot of time putting together. And we also have a package for calculating energy distance and energy tests as part of that. We have this sweet logo, which my sister Sophia made, who I probably on this call. And the data and everything for all of that is on scperturb.org. So using that, we try to benchmark how e-distance performs in different contexts and what sort of things it's robust to. So for example, energy distance is robust to asymmetric sample sizes. And we can show that on this data set from this is the same one where I just showed you that JAK-STAT pathway. We take for all of the perturbations in the data set, we compare their energy distance to the unperturbed cells. And we do that while changing the number of unperturbed cells that we use for the calculation. So way out here, we've used all of our cells to calculate. And then as we move further this way, we've dropped more and more cells from the analysis. And we've just done this once for each perturbation, because at each of these points, we've also gone in and calculated the energy test score, so whether or not the perturbation had a significant effect. And that's what we see on the right. So on the left, we see the distances. And we can see that the distance, as we decrease the number of control cells, once we have above about 15 control cells, the distances are fairly constant with some fluctuations. And then the significance as well starts to get stable, really stable around about 100 cells. So if we've got at least 100 cells in each of our populations, our distances are pretty stable and our significances are quite stable. So that's quite cool. And we did this for a lot of other parameters, which I will refrain from going through with all of you because it's really just the same plots over and over and over again. And we go oh, yay, it still looks good, happy about that. And a little bit of okay, it looks good within this regime, or if we have at least this many genes and that kind of thing. And one of those, which was particularly surprising to us, is that our energy distances were robust to the genes that were selected prior to computing PCA. So I told you guys a little while ago about how we reduce our data from this 20,000 dimensional thing down to much fewer dimensions. But we actually I skipped a step there, which is that when I'm actually doing this, before I even calculate the PCA, I dump 80% of the genes into the garbage and ignore them. And we do that for a number of reasons that are mostly it improves performance, but how you decide which genes to drop when doing that is sort of a complicated decision. And for this type of data, one of our questions was, okay, usually we choose genes which have a lot of variability before we do our PCA. But if we're choosing genes that have a lot of variability across the whole data set without taking into account which ones are distinguishing our perturbations, we might be losing some conditions where the genes that are perturbed are ones that are only varying for that one perturbation. So we tried two different methods of choosing genes. So there's a perturbation independent method, which is kind of the standard approach, where you use highly variable genes. And then we compared that against a perturbation aware method, where we take differentially expressed genes for each of our individual conditions, and then we pool those together. So in this particular data set, we took we ended up with about 2,000 genes using our perturbation independent method and 2,700 genes when we've augmented. And in the middle, there's some that were included in both. And what we found was that our energy distance is actually robust to those two different methods. So we're not dramatically losing how significant perturbations are by not taking into account these perturbation-specific gene changes before calculating, which we were thrilled to discover that, because the differential gene expression method is very computationally intensive relative to the other methods. So this means we can save on all of that effort and we get basically the same e-distances out. And now that we've decided that our energy distance works, we can make experimental recommendations. So if you come to me and you say, Tessa, I want to do an experiment and I want all of my perturbations to be distinguishable from our main data, and I'd want to make sure I sequence enough cells that I'm going to be able to tell if my perturbation worked. Because some of these are kind of, sometimes they work and sometimes they don't, and I want to know which ones are working. I can tell you, okay, as long as you get me at least 1,000 cells from each of your conditions, or sorry, not 1,000 cells, this is the how many sequencing depth ones. As long as you get me at least 1,000 reads from each cell, so within each cell, you check at least 1,000 pieces of RNA for what gene it encodes, that's going to be enough for me to tell you if your populations are distinguishable. And if we add more after that, for most modern sequencing experiments, it's not really going to improve my ability to answer that question. So that's what we've got here. And we did this particular one for a whole bunch of data sets. Okay, so that is the distinguishability project. And sorry, I'm just realizing I have no way to tell how long I've been talking for. Okay, fantastic, great, I think I'm on track. Okay, so we have a publication on this, which is going to be coming out in Nature Methods very soon. Pretty excited about that. So I told you we have the SC perturb package for e-distances. We've also incorporated the SC perturb, the energy distance and energy statistics, as well as a whole host of other distance measures and evaluation measures into this new package PertPy, which is part of the ScanPy SCverse Python single cell ecosystem. And then we've also added in programmatic access to our database via that package. So I think it's going to make it a lot easier for people to use this, which is pretty cool. And then the other thing I should note is that I've talked about e-distance and e-statistics just as a distinguishability measure or as a distance, but it can also be used as an evaluation metric. So if people are building generative models of single cell data, so if they're trying to predict what the gene expression would be in a system, or they've measured chromatin accessibility and they want to predict the RNA, you could use energy distance as a metric of how well you've done those predictions. And we've compared e-distance against a bunch of other evaluation metrics, and that's, I guess there's an in-progress workshop paper that got submitted last week, and we'll have a more final version soon. And Yuga has been taking point on that. Stefan has been also helping with that. And then Lucas and Yuga together are working on PertPy. Lucas is the PertPy chief who makes sure that we write good and maintainable code. So thank you, Lucas. Yeah, so that's what's happening on the distinguishability project. So we've done all of that. Even though we can say that cells are definitely different or definitely not different, there's still the question of what makes them different and when those differences matter and what we can do with that biologically. So I'm going to try to do a little bit in terms of how we answer that for the next part of the talk. And for this part of the talk, so a lot of the code that we used here was written up in Python by Yuga. And she also was a lot of great discussions. And it was co-advised by Chris and Debbie, as well as Linus Schumacher. And then Giuseppe works with Linus and was also sort of involved in this project and had a lot of good discussions with him as well. Okay, so we're focusing here specifically on triple-negative breast cancer. This is a type of breast cancer which is particularly difficult to treat. When I say it's triple negative, what I mean is that it doesn't express any of the receptors that are listed here. So that's estrogen receptor, progesterone receptor, human epidermal growth factor receptor. And the reason that that matters is because there are treatments that target these cells that express each of these receptors. So if you are lucky enough to have a cancer that has one of these, you can use treatments that are targeted to those, and survival tends to be better. Triple negative breast cancer also in general is more aggressive, can be faster growing. So we want ways to treat this that are more specific, and we want to understand why it doesn't respond to treatments and when it doesn't respond to treatments and what we can do to help it respond better. And when we're thinking about that, part of what we're thinking about is not just the tumor itself, but also the tumor microenvironment, or the immune cells and the other cells in your body that are around and within the tumor. And those cells are also controlling whether our tumor responds to treatment. So when we think about a tumor, like you might picture like, okay, it's this like sort of homogeneous mass of cells, but really there's a lot happening there. So it's got immune cells, it's got, let me get my little laser pointer again. Yeah, we've got immune cells, we've got like regular non-tumor cells, we've got fibroblasts. So these are cells that are making like connective tissue that are all over your body. And within the tumor, all of these are talking to each other. So they're expressing various receptors and listening to different signals, and they're expressing different proteins that are acting as signals. And this network of communication controls how cells are responding to the environment and what they're doing functionally. So for example, in triple negative breast cancer here, they've treated it with this drug called doxorubicin, which is a chemotherapy agent. And in this particular case, even though they've given it this chemotherapy agent, the cells are not responding to the treatment. And the reason for that is that the fibroblasts in the tissue, so even though they're not part of the tumor, they're emitting signals which are telling the tumor to ignore the doxorubicin, not take it up, not die, basically. And if we add in an anti-HMGB1 agent, so this is an antibody that targets the signaling factor that the fibroblasts are using to talk to the tumor cells, then the doxorubicin is able to kill the tumor. So by understanding how cells in the tumor microenvironment talk to each other, we can intervene and improve response to treatments. So that's the motivation for why we want to understand this stuff. Okay. So in this particular project, I'm focusing on a pre-treatment tumor data set. This is a published one. We're like going forward, Giuseppe and Luciana are working with some unpublished data. But for now, I'm going to talk about this data from Zhang et al. This was pre-treatment tumor samples that we analyzed here. They also, if you want to go read the paper, they looked at post-treatment as well. And these are patients where some of them received neoadjuvant chemotherapy, and some of them also received an immunotherapy drug. And after getting those drugs, some of them had their disease not change, and some of them had their tumor shrink. They didn't have a full loss of the tumor because in this particular study, they were looking at how the tumor changes. So people who didn't have a tumor afterwards aren't part of the study. But our forthcoming data does have some people who did fully respond. Okay, so this gives us 4 patient populations. We have people with stable disease in blue and green. We have people with partial response in red and orange. So you want to be in the red and orange group. And then we have 2 different drug groups. So there's really 4 groups here. So what we're trying to look at is we're looking for what predicts people who are not going to respond to treatment or who are going to respond to treatment. And we care about that because you don't want to give people a treatment that is just going to make them sick without benefiting them. And we also care about it because if we can understand why people aren't responding, we can potentially intervene and improve response in the future. Okay, so we've done, or they have done single cell sequencing on all of these patients' tumors before they got treated. And then we have cell type labels here, which are from the original study. And if we look at blue, we have patients who responded, and orange, we had patients who didn't respond. Just by eye, you can see that there's a difference between the patients who responded and the patients who didn't respond within each cell type. So for example, our macrophages down here are very clearly different between people who didn't respond and people who did. And we can quantify this difference using e-statistics, which I was just like we did in the earlier part of the talk. So here on the bottom, we have all of our different cell types. We have them colored by how many cells of that type were observed in the tumor samples. And then on the top here, I've labeled which ones had a statistically significant energy distance between the responding and non-responding patients. And what I did here is I pooled the two groups, but evenly. So like half of the cells are from people who got one of the treatments and half of them are from the ones that got the other treatment. And we can see that a lot of our cell types are distinguishable, but what we want to know is not just, are they different, but what are they doing that's different? And are they working together in a way that we can access those changes? So to do that, we've brought to bear another mathematical method, which uses matrix decomposition, similar to principal component analysis. So what we're seeing here is we have our 2D projection of our data. We have two different cell types. I've just called them A and B here because it's just to build a little understanding. We have each point here is a cell type average for a single patient. So this is for cell type A, our pink patient is like chilling out here with, and then our red patients are up here. And with cell type B, pink patients down here, red down here. And mathematically, what we're looking for is we're looking for correlations that are seen across cell types and across patients. So directions where as we move along that direction in cell type A, and we move along the corresponding direction in cell type B, patients that have a low score for cell type A tend to have a low score for cell type B and vice versa. And mathematically speaking, we solve that but we find that by solving this optimization problem, which looks extremely similar to the one that we were looking at earlier for PCA, with a couple of changes that I'll tell you about in a second. And what that gets us is it gets us these correlated, what we call multicellular program or MCP scores, where for cell type A, MCP1, cell type B, MCP1, they're correlated with each other. So we have our pink values down are low for both, our red patient is high for both, and they move along this gradient together. And the optimization problem here, very similar to PCA. So in PCA, we take our XTX here, and they're both onto the same U. In our multicellular program, one of them is going on to the vector corresponding for that cell type, the other one is going on to a vector corresponding to the other cell type. And then we've also, instead of setting the size of the vector equal to 1, we set the L2 norm has to be less than or equal to 1, and the L1 norm has to be less than or equal to some constant, where the constant controls how sparse our resulting outputs is, or how many cell types are contributing to the data set. And this is very close to equivalent to what's called sparse PCA, which is a modification of PCA, where instead of having, you have fewer components per thing, basically. Okay, so once we get out our multicellular programs, we want to ask what genes are contributing to those multicellular programs. And we are using a heuristic for the sake of this talk. In the long run, we'd like to compare various other methods for doing this. And in my thesis, I went through a couple of other methods. The heuristic that we used here is we identify cells that have low MCP score and high MCP score within each cell type, and then we just directly test genes that tend to be higher or lower in the two groups, and we use those as our MCP related genes for trying to interpret what's happening in these multicellular programs. And if we do this on a breast cancer atlas, so this is a different data set where they've sequenced a lot of tumors from people who have varying kinds of cancers, as well as people who have like precancerous tissue. And then we look at our first multicellular program, so the direction that has the most significant change. And we look at two of the cell types involved in that are cancer-associated fibroblasts and tumor-associated macrophages. So these are the cells that make connective tissue in the tumor region, and these are cells that these are white blood cells. And the fibroblasts and tumor-associated macrophages, we find that when fibroblasts are expressing factors related to proliferation and glucocorticoid response, the macrophages tend to have changing levels of these calcium channel regulators. And it's not totally clear like what that means in terms of what's going to happen with the tumor, at least not from this data set. But we do know that cancer-associated fibroblasts regulate tumor-associated macrophages. There's a large body of existing work showing that these fibroblasts can reprogram immune cells in the tumor microenvironment. So we think that this is capturing an example of fibroblasts influencing macrophages. And in our cancer treatment data set that I told you about, when we run this algorithm, we get out a number of multicellular programs, and this particular one seems to be predictive of treatment response, where we can see that our blue and green treatment responders have a high, or treatment non-responders have a higher MCP score than our treatment responders. And a few of our patients who had lower MCP score and didn't respond to treatment, they only received the chemotherapy. So it's possible that these are patients where if they had received also the immunotherapy drug, they might have had a stronger response. And the genes that we see involved in this, in our memory B cells, there's IL-7, along with many other genes, is higher in our non-responders. And the receptor for IL-7 is expressed in the T cells in both responding and non-responding patients. So we think there might be a shift in the amount of IL-7 signaling between these cell types. The T cells in these groups are expressing the AP1 transcription factor at a higher level in patients who don't respond. This is a transcription factor that's known to be related to cancer response, or to treatment response, but it seems to depend a lot on the specifics of the treatment and the specifics of the cancer type and other things like that. So it's a little hard for us to tell quite what's happening here, but it seems to be maybe interesting. And in all three cell types, in the non-responding patients, there's a much higher level of heat shock proteins, and that could be indicative of stress response. So this is sort of cool. So it gave us a view into what's happening, and we can tell these kind of stories that weren't really possible to tell without using this mathematical method. But can you yeah, thanks. Okay. But it's still limited in that we only have our one data set. We're drawing conclusions that are or we're trying to draw these kind of causal conclusions, or these answers about oh, we think our memory B cells are sending a signal that our T cells are receiving, but we have to make a lot of we're inferring a lot of leaps as we're doing that. So really, to kind of say something more about whether this actually matters, you need interventional experiments, or at the very least, we need more data and more patients. And the other thing that would be very cool to have would be spatial information. So are these cells actually close enough to each other in the tumor to be having a chemical conversation? And that's something that we are going to have very, very soon. So Giuseppe and Luciana have been hard at work on this. Our collaborators, as part of the LEAP Delta tissue team in London, have been gathering tumor samples from hundreds of patients, and they've been doing a lot of different spatial analyses on it. This is just spatial proteomics. They also have spatial transcriptomics on adjacent slices, as well as some other methodologies. And so from the proteomics, we have these, we can look at our slide, we can see all of these cells, and then we try to assign cell labels to them. So using the same kind of methods that we use for RNA, instead we try to look at these pictures and say which cells are present and which ones are near each other. And then once we know which cell has which label, we can try to quantify which ones tend to be closer or further away from each other, and gets us a little closer to being able to tell what kind of communication is possible. So that's where Giuseppe and Lou are on that work. So that's coming down the pipe. And we also have some RNA sequencing and single nucleus RNA sequencing that will be coming in the future from that, from a lot more patients than we have in the little pilot that I've just showed you about. So it should be very exciting to see what happens next with that. And then the other thing I wanted to note is the dialogue package that we used was written in R initially, and Lucas and Yuga, mostly Yuga, have re-implemented that in Python as part of this PertPy package, which I mentioned earlier. It's going to include the energy distance stuff. And they've also been working on a new solver, so with better robustness to various input parameters, improving the gene set detection methodology, and of course, applying it to new data as it comes in. They're also part of this collaboration with the experimentalists and everything else. So yeah, that's what we've really been doing here. And I hope that this talk has shown you a little bit of how I've been working to try to make meaning from this single cell RNA sequencing data, and how we really try to get interpretable and usable results out of these big messes of very interesting data. And going forward, what we want is to move beyond just naming cell types or distinguishing cell types and towards understanding what drives cell state transitions. So not just is our tissue changing and when is our tissue changing, but what dynamics underlie those changes, and moving forward with all of that. So that's what I wanted to cover today. So thank you all so much for listening to all of that. And thank you to, okay, I'm just going to start in on my many, many thank yous. So these are all of the people that financially supported my work during my PhD. So Delta Tissue funded all of the work that was in this talk. And prior to that, in my PhD, I was supported by the Chan Zuckerberg Initiative and the NIH and a fellowship at Harvard. Okay, thank you so much to Chris and Debbie. You guys have been really fantastic. I joined Debbie's lab, I guess, much earlier in my PhD, and Debbie has just been so supportive of me, like very much from the beginning, and really in my ability to like, just supporting me being able to make the choice to do whatever science I want to do, regardless of health issues and regardless of everything else. And it really means a lot to me. And really giving me the freedom to explore things intellectually. So this work is very different from what most of the Marx lab does. And I was very lucky to start working with Chris later in my PhD, who works more on this kind of stuff and has been incredibly intellectually supportive and helpful with all of the going through the paper process and everything and building a really fantastic collaborative team. It's been really fantastic to work with both of you. Thank you so much. And I had a fantastic committee through various iterations. I guess all of the work that you heard about was really the last 2 years, and I went through some other, I guess, lives of PhD earlier with different committees and everything, but everyone's been really supportive. Alon has been along for the entire ride, so thank you. And the biophysics chairs have also been fantastic. I think I managed to list all of them. I'm not actually sure. There's been a few. Okay, Michelle's nodding, so I guess I did it okay. Martha in particular has been really helpful this last year or so. So thank you. And I wrote Jim twice on his, whatever, it's fine. I couldn't have done any of this work without my fantastic collaborators. Stefan in particular has the last 2 years, we've been really I don't know, intellectual partners on a lot of work. And I'm incredibly grateful to him for all of his help on everything and just doing great science together. It was very weird putting together this talk and talking about the work and everything and doing it without having Stefan just next to me for all of the meetings so that we could answer all of the questions together. So that's been kind of an adventure. Yeah, so Stefan, along with Niels, Augustin, Judy, Torsten, and Jake on that first project has been great. Earlier in my PhD, I worked on asthma for a while, and that was with Tanya and Katie, who are doctors at the Brigham, and they were also great. I told you about Lucas and Yuga already, and Linus and Luciana and Giuseppe, just really amazing people, and it's been so fantastic to have them. Okay, I've been housed in the Marx lab for my PhD. You guys are the best 2 rows or whatever, the audience here. Yeah, they're just a really, really great group. Debbie, I don't know how you're so good at hiring people, but it's just such a supportive and wonderful environment where I always felt very safe taking intellectual risks and never felt like I couldn't ask a question because I'd feel stupid. No one cares. It's so great. Kelly is not in the photos, but Kelly is amazing and has been so supportive and left the lab a while ago, but still lets me call her pretty regularly. So thank you, Kelly, you're the best. Everyone's the best, whatever. Okay. So I'm part of the biophysics program, like I mentioned, but I'm housed with Debbie and Chris in the Department of Systems Biology. I tried to find photos of the program or the department, and it turns out I don't take photos of scientific context. Deborah is my friend from the Systems Biology department, who we co-taught before I joined Debbie's lab, and that's us when we were teaching along with our other co-teacher, Peter. She brought me into the systems biology, dev biology, single cell reading group, which we had for a few years there. And that was really super helpful for my intellectual development and project development kind of stuff. So thank you all for that. In biophysics, literally my only photo of anyone in biophysics is this corner of Kevin's face. So thanks, Kevin. And thank you everybody else from biophysics for being so fantastic and all of our amazing biophysics retreats in Maine. I'm sad that I couldn't go to the one a couple days ago, but I was a little busy. Here's some of my fantastic roommates, past and present. So it's been a great intellectual home. I would not be here without my union. Rachel, you're my favorite. Okay. Yeah, so I'll talk a little more about this, but I have a lot of health issues, as everyone in the lab, and probably everyone who knows me knows at this point. in 2019, I gave a speech on the quad while we were on strike, in which I talked about the fact that I am able to work with these fantastic physicians at the Brigham on my research, but our health plan at the time meant that I couldn't get medical treatment from them. And because of the union and everything that we've done, like I have, like that's totally changed now relative to how it was when I started. And I'm so grateful to everybody who made that happen, along with, I guess, me. So, you know, go team. And thank you, Rachel, for, you know, wearing your great hats and all that. Okay, so I would not be here without the support of my incredible medical team. There's me and Amelia, my sister, at the hospital in April. I went to the emergency room for my heart, and then I did not come back to Harvard since then. This is my first time, you know, sitting in a chair and not working from my bed in about 6 months. But I'm here, so I'm pretty happy about that. And I'm still trying to figure out my meds and everything, but it's been, I feel really lucky to have had the support that I've had and have the financial resources to have all of that too. So big thank you. And thanks to Amelia and Shen for coming to the ER with me. Love you guys. Okay. Oh, I'm going to start crying. Okay. I have so many really, really amazing friends who have been with me through a lot. Yeah. So my high school pals, my like summer research high school, like I've kept a lot of friends for a really long time, and all of you are like really amazing people, and I feel really, really lucky. You guys, my like grad school BFF who finished a year ago because she's more efficient than I am, but I think she's on this call. I love you. Okay. Yeah, I have so many fantastic friends. That's us doing a Python B. One time I won a programming competition against software engineers, and I'm going to be proud about that for the rest of my life. This is us more recently, less recently, just, you know, I'm really lucky. Yeah, this is my girlfriend, Shen. You've been really, really good. Especially, like I said, the last 6 months or so has been really hard, and you have just been like such a trooper. And I'm really, really lucky to have you. And I'm excited to, you know, keep hanging out and all that. And, you know, it's pretty good. Thank you. Love you. Okay. Yeah, I have a huge and amazing family. A lot of y'all are on Zoom right now, I think. Yeah, just you guys make me laugh so much. Like I think in half of these, I like laughed so hard I was crying by the end of it. I did not, yeah, we've got Michaela doing some charades here. Like I was like sobbing laughing at that. So just, I love all of you. It's so great. Okay. And my family has been so wonderful. Mom, you're on Zoom because you're sick, but I hope you feel better soon. And I'm glad that Dad's here and everything. My parents have been so supportive through all of this. Even though some of the time they like didn't understand why I was doing certain parts of it or why I'm still here, but I'm still here and I did it, and you guys really helped a lot. Amelia's here today. And my other sister, Sophia, she studied science illustration in school, and she made our logo for the SE Perturb project. So she's super awesome. My wonderful grandparents, I think someone got grandma on Zoom. So hi, grandma, if you're here. My other grandparents aren't with us anymore, but they were always really, really supportive of me. Rhoda in particular spent a lot of time on the phone with me while I was figuring out what lab to join and going through the early years of my PhD, and it has meant so much to me. So just, okay, cool. Okay. Thanks, everybody.
Take some questions. And is somebody, if anybody's got one from Zoom, is somebody overseeing that? Anybody got any questions? You were just so amazing at explaining absolutely
thank you.
everything.
Tessa, did your awesome drawings here was like
Me. Thanks. Yeah, I did not have Sophia do them. I did them.
Great, that was my question.
Well,
So what should we be measuring?
Oh my gosh, everything. No, I really like, I like RNA a lot. It's been fun. I think like, yeah, no, I mean, you know, great job on that chip, you know, it's been really pretty impressive. No, I think we should be getting more out. I'm excited about the, like, I haven't done any of the RNA velocity stuff, but like making, like actually getting more out of the reads. would love to do an entire project trying to get the like mutation information out of the RNA when you're doing the sequencing, but limited time. I think the like multimodal stuff that's coming out is very exciting, but it's what ends up being useful, I don't really know. What I'm definitely finding as we've been trying to do the spatial stuff is that some of the skills transfer, but like a lot of it really doesn't. And to some extent, I feel like it's good that we keep working with RNA even when the new methods come in, because like at least we are a little bit closer to having tools to make sense of stuff. I feel very hacky doing the imaging analysis so far. Yeah.
Anybody else? Anybody on Zoom? Ask anything? Oh my God, you're so well explained that.
Thank you. Or they didn't follow it, so there's too many questions. Yeah, pretty.
Actually, we're
Okay.
you explained like the polyenergy of like, specific was robust, like how you threw away your genes. Do you know mathematically why that is, why it's robust?
Nope. Okay, so the actual answer there is probably that the genes are so correlated with each other that it doesn't really matter. And that's like part of why we end up throwing a lot of them away anyways. Like you have 20,000 genes, but the amount of actual information is much less because they're in these like regulatory networks, and the structure of all of that, we think means that one thing we didn't test, but I'd love to, is just grabbing like random sets of genes rather than filtering them in any way and seeing if that gives you something similar. And honestly, my guess is that if you took 2,000 random genes, you'd get a pretty similar result. Yeah, sure.
Do you want to read out the question? Can you say who it is? Or?
Yeah, we've got, okay. Zeke is trying to ask something. Zeke, can you talk or no?
I think so. Can you hear me?
I can't hear him.
No.
He's unmuted. I don't hear it.
Well, just read out.
My speaker is on. I don't know. I can't hear anything. Okay, Zeke, can you type it? Sorry, man. See. Okay, so you can type it. Hang on. I'm just seeing if we got any other ones typed in, but this isn't. Alan, that is not a helpful question. Alan said, "How did you do such a great PhD?" I had a really great team. Yeah, that's our Alan. Yeah.
Alan.
Okay, Ruben has a question. We've answered that one.
I have a question about the, you showed like the plots where there was all of the I think S sequence of data sets, and you pulled 3 where there was no like significant.
Yeah.
So how do you what happens if you integrate all of them in one? And if the instance specific still able to distinguish with error or what this
So if we have if we've pooled a bunch of perturbations together, how well does it work? Yeah, so I haven't done exactly that. Maybe a sort of similar thing is how we perform if we've combined across batches. So we did one analysis where we took cells from different patients and pulled them together in a case where the patients were very different from each other in the underlying distribution. So I don't have any, I'm not sure quite how to answer the exact perturbation question, but at least in that case, as long as we took approximately even numbers from the kind of very different from each other ones, we could still get a measure of how different the overall pool was. So yeah, I think if it gets too messy, it becomes difficult, but you still get some measure of distance. There's some more complex measures for distributional distance that might perform better in some of those cases too.
It's a nice question.
Yeah, it is. It's just it's a hard one to.
The experiments would be, and then what we use so they can interesting.
Yeah, okay. Oh, and Zeke asked, how computationally intensive is e-distance relative to established methods for differentially expressed genes? Yeah, okay. So e-distance is way less computationally intensive than just a DEG checker. So and you can use eDistance, it's integrated with Seurat, by the way, Zeke, so you can use scperturb R if you want to try it out. It's asking a much simpler question than differential gene expression, because for that, you're trying to answer for each of my 20,000 genes or whatever, which one is different. So you have to run a lot more tests, whereas the eDistance is equivalent to testing just whether one gene is differentially expressed or not. So it's a lot faster. It's also much faster than the Wasserstein distance and MMD distance and a lot of other sort of conventional distances. So that was part of what we liked about it, is that it's pretty quick. Our implementation is like medium quick. The Python one, we've done some like pretty nifty accelerations, so it's really very fast. The tests are still somewhat slow because just you have to run a lot of them, but it's still faster than Wasserstein by a lot. Yeah.
Okay, I think that we could round off now and then go and ask the questions. The next day, thank you very much, Tessa. Now it's time to quiz you.
Yep.